we are so happy to mark the start of our bioengineering seminar series for spring 2016. As you are aware, starting from this semester, we have our interdisciplinary PhD program. And we plan to have a seminar every week. So you are invited. Today there is a delay in ordering the pizza, so we will serve the pizza after the seminar. We plan to make it before the seminar, but we are sorry for this. Before we start, I'd like to thank Dr. Malik for accepting our invitation to be our first speaker in this seminar series. Dr. Malik, he is Assistant Professor of Medicine Division of Medical Hematology and Oncology. Also, Dr. Malik is a member of Brown Cancer Center at University of Louisville. Dr. Malik graduated from Biochemistry Department at University of Louisville, and he did his postdoctoral training at Brown Cancer Center. Dr. Malik's research interest is to develop multifunctional nanoparticles for imaging. So we are working with Dr. Malik, and we are so happy to start this seminar.
Thank you, Dr. Baz. Sorry for the inconvenience, some technical difficulty. Now we are on the path. So today my talk is the new generation of cancer medicine. So we at Brown Cancer Center try to develop new medicines for cancer therapeutics. The problem with the cancer is that we don't have any good chemotherapy that can cure the cancer. And we are still finding the new ways how we can make these drugs more not harmful, and they are being used regularly. So today I'm going to talk about synthetic nucleic acid aptamer guided nanotherapy for the breast cancer. So I divided my talk into 4 different parts: discovery of aptamer AS1411, development of AS1411 gold nanoparticle, and its potential use as a radiosensitizer, and also AS1411 gadolinium conjugated to gold nanoparticle as a radiosensitizer, also as a contrast agent. So what are the aptamers? Aptamers are the synthetic oligonucleotides. Either they are a sequence of DNA or RNA, about 15 to 40 nucleotides long. They bind to their target very specifically due to their shape recognition. And they also contain some secondary structure. And that is very important because according to the protein secondary structure, these DNA molecules have to fit the target. Sometimes they may form hairpins and G-cotton. As here there are 2 examples of neomycin aptamer. This is the RNA aptamer, and it binds to a neomycin and it forms the hair rope. Whereas the thrombin aptamer, which is a DNA aptamer and forms the G-cotton, and it also binds the thrombin molecule. So what are the advantages of aptamers? They bind to a molecular target with a very high affinity and specificity. And they can be easily modified, conjugated to fluorophores, radionuclide, toxins, and even nanoparticles. And usually their limit is extracellular or cell surface target. It's easier for the aptamers to bind on the surface rather than going into the cells. Advantage over antibodies. They can be easily produced. There is no animal needed. They are chemically synthesized. There is very little variation between batch to batch when you synthesize it. And they are stable at a room temperature for a very long time. So they have a very long shelf life. And the best part is there is little or no immunogenicity in animal model or even in humans. And they are smaller, and that's why it is very easy to put a higher density on nanoparticles. The only disadvantage these aptamers have, they have a very short half-life in the circulation. They are secreted away within few hours after injection in humans or in animals. So, the discovery of AS1411. This was discovered by Dr. John Miller, Paula Bates, and Dr. John Trent in Cancer Center when they were at UAB. And they are looking for triplex forming oligonucleotides designed to inhibit the gene expression that are important in prostate cancer. And what they found when they are analyzing thousands of different sequences, that there is one sequence, is a G-rich DNA sequence that causes selective inhibition of cancer cells proliferation, which is unrelated to its triplex formation to the DNA. So this is just by chance. Usually the aptamers, they are formed by a process called Celex. So this is being discovered by chance. It's not by Celex. So this is the sequence of G-rich AS1411. It's about 26 base pair, unmodified phosphodiester oligonucleotide. Previously it has been given many different names, AGR100, GR26B, and so on. And it forms a very stable G-cortex structure, which it makes resistant to the nucleus in the serum, and work as an aptamer by targeting a protein that is present in the nucleus. It's called nuclein, which is also expressed on the cell surface of only cancer cells, not the healthy cells. And it is, I have already said, discovered by chance, not by Celex. Why AS1411 is interesting? What's the unique? It forms a very stable G-cortex structure. It has an anti-proliferative activity in a wide range of cancer cells. And the other important thing is they are very cancer cell selective. And they also deplete cancer stem cells. Efficient cellular uptake compared to other oligonucleotides on the market. And this is the first aptamer entered the clinical trial, human clinical trial. So what's the mechanism of this AS1411? It binds to nuclein, which is present also on the cell surface of the cancer cell. Nuclein is a very multifunctional protein. It shuttles between nucleus to cell surface and then back. Somehow it is being stuck on the surface of the cancer cell, but not on the healthy cells. So that makes it very unique that AS1411 binds to the nuclein, and then by the process of endocytosis, micropinocytosis, and many other processes, it's being taken up by the cells. The anti-proliferative activity is it arrests the cell cycle. And it also blocks the DNA replication and inactivates in a kappa B pathway. And induction of many tumor suppressor genes in the cell. And reduce BCL2 expression, which is in the pathway of apoptosis. Recently from our lab, we found that AS1411 hyperstimulate micropinocytosis to induce the cell death. How it did? As you can see from this model, AS1411 binds and uptake by micropinocytosis in cancer cells, also in normal cells within the first 2 hours, and this is nuclein dependent. After 24 or 48 hours, what happened? It is stuck inside the cells of the cancer cell, as whereas in the normal cells it is being pushed out, efflux. And that is how the cancer cells, they retain the AS1411, which is very unique property of this. That's why it has been uptaken by the cancer cell, not by the healthy cells. So there was a first human clinical trial of AS4 at James Brown Cancer Center. What is the experience of the clinical trial? About more than 100 patients treated with no serious side effects. Some evidence of clinical benefit. Response is very low, but durable and dramatic. Potential result of this low response rate is poor pharmacology. As I have said you before, it does not retain in the blood circulation. It is excreted out very fast from the blood circulation. And low potency, because to kill the cancer cells, you have to maintain a certain threshold of concentration in the serum. But that was not achieved in human clinical trials. And also, still we are trying to find that what's the exact mechanism of AS1411 to kill the cancer cells. There are many, but not definitive. And we still did not understand that how and what's the reason it has this so dramatic effect on the cancer cells, not on the human, normal human cells. And there is no biomarker we can able to find that how it's working. Anyhow, so we came up with a solution that how we can retain its efficacy and potency and higher circulation rate in humans. So we came up with the nano formulation. So our team, myself, Dr. Bates, and Dr. Martin Tully, and Dr. Don Miller, we came up with the chemistry of conjugating AS1411 by a thiolinker to gold nanoparticle. And this is the simple schematic how we conjugate the gold nanoparticle and then how to take off the AS1411 from the gold particle to characterize how many particles and how many gold nanoparticles are being attached. So the characteristic of this complex is about it is 5 nanometer solid gold core, and it has about 42 ligands of AS1411 per particle. The total diameter of complex is about 32 with a zeta potential of -26. So our preliminary results indicate that it dramatically increased the uptake by the cancer cells and up to 50-fold increase in the potency against cancer cells comparing to the healthy cells, and maintains cancer selectivity, that it has no effect on the normal cells. Selective and antigen accumulation in tumor in mice model, and anti-tumor activity in mice model and without any evidence of toxicity. So Dr. Don Trent, he have done some modeling and find that what are the physical characteristics of our data, that gold nanoparticle of 5 nanometer, which has about 3,900 atoms of the gold, and on the surface is about 40 to 42 AS1411 molecules can fit. So he predicted that model and it fit with our physical characteristics. And it is stable in very many solvents we tested, like DMS, DMF, and also stable in mediums, culture mediums, FBS, fetal bovine serum, and in water, up to 7 days, and even longer than that. So it is very stable when it has been uptake by the cells. As shown here by silver staining, you can see these black spots. These are the gold nanoparticles, and they are conjugated with high fluorescence molecule and they overlap. So it shows that they are very stable and intact in the cells. And in vitro cell uptake study in MDA and MCF7 cell line, you can see here, Cy5 level AS1411, 10 micromolar, comparing to conjugated to gold nanoparticles. 50-fold decrease in the concentration of oligos, 200 nanomolar. The uptake is dramatically increased, and it retains in the cancer cell, and it can have a very potent effect on the cells. So here we are showing that it has selective uptake comparing to non-malignant setting, which is MCF10A, comparing to MDA. As here you can see in silver staining, they are showing the same thing here, comparing to our control and untreated cells. So when it takes up the AS1411 GNP, can it kill the cell, or what effect it has? So in our cell proliferation assays in MDA cells, which is the triple negative breast cancer, estrogen dependent MCF and MCF10A, which is non-malignant cells. You can see here AS1411 dramatically kills the cells as the concentration goes up. The IC50 value here is about 100 nanomolar, as compared to AS1411 about 3 to 5 micromolar range of concentration. So it dramatically increased the potency of the AS1411 conjugated with gold nanoparticles. But there is no effect in the non-malignant cells. We have used another model from the rat, which is a liver model, and it shows the same what we have seen in breast cancer cells. This is just to show that these are all cancer cells, except this MCF10A. When you use AS1411 alone, the concentration is 4, 5, 5, 1.5, 10, 4. And when you conjugate it to gold nanoparticles, how the oligo concentration goes down. Less than 200, less than 100, 300, 80 in nanomolar range compared to the micromolar range. So how it kills the cells? So we done some clonogenic assay and find out that it's showing here GNPS 1411 at 200 nanomolar almost kills about 50% of the cells, and the process by to do that is by necrosis. Whereas 50 times more AS1411 have only 27% of cell death, also by necrotic, but this is like a huge increase in cell death compared to AS1411. So in animal biodistribution, when we inject by retroorbital injections these gold nanoparticles, within 6 hours it is all over the body. As the time passes, after 7 days, you can see it's only accumulated in tumors. Whereas GNP CRO, which is our control, it just wash away. And looking at the organs from these animals, you can say there's a higher uptake in the tumor, a little bit in liver and brain, but not in other organs. So this is very encouraging thing for us. And we have repeated this experiment and shown that in almost the same results we got. There's a higher uptake in the tumor as compared to the other organs. So what does it mean? Does it kill the tumor or not? So we find that when we injected about 100,000 cells in a xenograft model of the animal, AS1411, and this is GNP. It completely abolished the tumor in the mice xenograft model. So next time we increase the number of the cells to the million cells, and we saw there is a more profound effect of AS14 GNP compared to only AS1411. That means it is a very good drug that could treat the cancer, and it totally abolished the tumor. And I have not shown the data here. We keep these mice for another 2 weeks, and the tumor does not come back. So that's a very encouraging thing too. This is another model, neuroblastoma, which is a very deadly childhood disease. And here we are also showing that conjugating to AS1411 to GNP, it totally abolished the tumor growth in these mice. And we have not seen any toxicity by gross anatomy, looking at these organs, and also monitoring the body weight of these mice. So the next topic I will talk about, can we use this GNPS 1411 as a radiosensitizer? So this is a project we started last year with Dr. Neil Dunlap, Dr. Anthony Palabes, myself, and Martin. So almost like 95% of breast cancer patients or all cancer patients, they have to receive some type of radiation therapy, either alone or with combination with chemotherapy. If they are able to control the local region of these primary tumors, which is very much correlated with the overall survival of unresectable tumors. Radiation therapy can cause acute or chronic side effects. Why? Because to kill the tumor, you need a very high dose of radiation. And it has produced a lot of bad or very bad lesions, burning around the skin, sometimes hair loss, memory loss, infertility, you name it. But on the market, there is no approved radiosensitizer. Currently, there are some chemo drugs they use, like 5-FU and cisplatin in combination with radiation therapy too. So the unique property of the gold particles as a radiosensitizer is because it absorbs 3 times more than iodine. It can increase the donor enhancement factor more than 5. And it has a longer blood circulation time than a small molecule like iodine. Gold nanoparticles can permeate into the leaky angiogenic endothelium and enter into tumor, which is EPR effect, enhanced permeability and retention, and capable of evading immune response. Low toxicity and low osmoticity even at a high concentration. Effective tumor targeting with surface ligand like antibodies, peptides, in our case, aptamers. So it can be used as a multifunctional gold nanoparticles. And the gold also has been used as a very good contrast agent in X-rays and CTs. And that will be helpful in looking at the biodistribution and planning for the therapeutic before the dose is delivered to the patient. So when the X-rays hit the gold nanoparticles, there are many other functions happening. Most importantly, autoelectrons, which is escaped from the outer shell, from the inner or the outer shells of the gold, and they produce a lot of damage to the DNA, and also produce a lot of reactive oxygen species, and also damage other proteins and lipids in the cell. which also increase the dose localization and enhance the radiosensitization. So on this principle, we said, okay, let's do, because when the gold particles are being delivered, if they are not targeted, they can go into the tumor, but they do not retain in the tumor bed. So you need a target molecule to keep these gold nanoparticles in the tumor bed. So the strategy we attached the AS1411 with the gold nanoparticles and deliver to the cells and extrude them. Because of AS1411, these nanoparticles are retained inside the cell, and when X-ray dose is given, there is a lot of DNA damage happens, reactive oxygen species produced, and there is a damage of lot of cellular component. Whereas the healthy cells, there is a minimal damage because these gold particles reflects from these cells. You can imagine that one 5 nanometer gold nanoparticle, which has about 3,900 atoms, and when X-ray hits, it generates a burst of outer electrons, and it can fatally kill the cancer cells. To show the proof of principle experiment, conducted one experiment with breast cancer cells. This is called a clonogenic assay, where you take the cells, you treat them, and then you, after different time points, treat with different radiation dose. So here you can see at a very minimum 100 centigrade, GNP, it kills the cells, whereas GNP AS14 completely abolished the cells. Here you can see the surviving fraction when you have a GNP and when the no. So it's about almost 2-factor increase in the efficacy of GNP in killing the cells. So we also evaluated how much DNA is being damaged. So phosphorylated gamma H2X is the indicator of DNA damage. And you can clearly see here, there's a lot of red color in the cells in the nucleus. That's damaged DNA in the cells. So we find that, okay, let's see where the particles are. Either our particles are near or around the cells. And we did an electron microscopy to show that all these nanoparticles are accumulated in the perinuclear location. And that is where we want our particles to be, to inflict a lot of damage to the DNA. This is the same experiment but in other cell lines in the lung cancer, and showing the same that when you have no radiation and with 100 centigrade, about almost 80% killing of the cells. So that's proof that we can use the AS1411 as a radiosensitizer. So another project we started just a couple of months ago, and it will go into next year. Dr. Malik, he is a diagnostic radiologist, and Dr. Aman Albaz, Martin Turing, Paula Bates, and myself, to make these particles as a theranostic. How can we use these particles as an imaging agent, also as a diagnostic, plus as a therapeutic? So Rajat Chauhan and Dr. Marcus Tully came up with this design where we have this gold nanoparticle, AS1411, and added gadolinium, which is an MRI contrast agent. And to see that whether we can use this particle as an imaging agent in MRI, CT, and can we use this particle also as a radiosensitizer, and also AS1411 as a targeted agent, so it can take the particles to the tumor bed. So the good part is that we have to buy the gold particles from the commercial source, which is very costly. Now in Dr. Martin Tully's lab, we are able to scale up the process, and they can make a very high concentration of gold nanoparticles conjugated with oligos and gadolinium. So that reduces the cost of our experiments and research. So the characteristic of this particle has a 5 nanometer gold core and about 13 ligands. We figured out that these are enough ligands that can carry the gold particles to the tumor. And the total diameter is about 13.45 nanometer with a zeta potential of minus 30. Because of gadolinium, I think that reduces the zeta potential, but it does not have any effect on targeting the tumors or in tumor cells. So we want to, and they also optimize the condition for gadolinium computation by Zenol and also EDAX, and find about almost 28 to 23 molecules are present on each nanoparticle. So we want to tell that whether it can make any difference in the uptake of AS1411 gadolinium gold nanoparticle, and we find that it still retains its activity and uptake in the cells as compared to our control CRO gadolinium GNP. So we done some preliminary data, and this is from CT. You can see the increase in house field units about 227 comparing to ES1411 only GNP. As we added the gadolinium, it increases its contrast compared to all our controls. So this is very encouraging, and we are still repeating this experiment, and in animal model, hopefully soon we will start to prove this proof of concept. And also with the help of Dr. Aman Albaz, we have done some MRI images, and we find that it has dramatically increased the relaxivity at both 9.4 and 3.0 Kelvin. So this is very encouraging that we can use our molecule for the contrast agent in MRI and for CT. So we want to also see that whether either we have the same effect of radiation on these particles or not. And we find that it kills the cells at the same radiation dose of 100 centigrade as ES1411 with GN. So it does not lose its capability to kill the cells even after adding the gadolinium on it. And this is just to show that there's a very higher and profound effect on the DNA damaging of this molecule comparing to our control. So this is very encouraging that we can use this molecule as a diagnostic, as a therapeutic, and also we can use this molecule to deliver different drugs to the tumor. So in I have shown you GNPS 1411 is highly stable and robust cancer targeting nanoparticle, and it stands alone as a therapeutic, radiosensitizer and diagnostic molecule, and it increases up to 54 in its potency and maintains cancer selectivity even after adding gadolinium on it. Anti-tumor activity in animal model without the evidence of toxicity. Whereas the GNP gadolinium AF1411 complex, it also has a very potent radiosensitizer activity at a low dose of radiation. So that is very encouraging. It has a better log T1 contrast agent for MRI and CT. superior relaxivity values compared to multi-Hz, which is the standard in the clinic. And it has the potential of theranostic agent as an image-guided nano radiosensitizer agent. So AS1411 future and our priorities are, we are still working on the long-term storage of these gold nanoparticles. We don't know how much longer we can store it and how much potency it have after a year of storage. So we are working on that one. We are also working on animal models to see its PKM toxicity, and also identifying the optimal dose and scheduling time. And we need to do some more experiments in different animal models. And yes, we need to find what is the mechanistic behind AS1411. Still we don't know what is the real mechanism, how it kills the cell. The good part is that at University of Louisville and Brown Cancer Center we have an advantage because we have a very good group of G-cartridges oligonucleotides who developed initially the AS1411. And we have a very strong bioengineering department and facility here at the University of Louisville. And we have a clinical structure and experience and institutional focus on translational research. And we have a support for this project, and we have a very good, strong intellectual property position on these nanoparticles. So these are some of our collaborators, past and present, lab people in our lab, Dr. Martin's lab, Dr. Chin's lab. We are working very close with all these are collaborators, and these are some funding sources we got money from to do our experiments and research. And that is all. Thank you. If there's any questions.
So we'd like to thank Dr. Malik for his good presentation. And now we are go to the audience. If they have some questions, they can ask Dr. Malik. Any question?
How did you actually end up choosing, like why did you choose gold as opposed to other elements? Like how was the process?
It's a good question. Why we choose it? Because for the surface modification, gold is very easy to tunable, and you can make a different size, and you can do a lot of chemistry with the gold as compared to platinum or silver. So we find that working with the gold is easier than the working with other metals.
Any other question? For me, I have a question. Like, because I worked with gadolinium before, and I know sometimes the people from Louisville didn't like it because it has some bad effect on kidney. What you are doing can be integrated with any other contrast agent rather than gadolinium.
We can do a lot of modification. Here what we are doing that the quantity of gadolinium we are using is far less than what in the clinical use we are doing. And we are still able to get the same contrast as at the higher concentration of gadolinium alone.
Yes.
So that makes it a little bit unique about this molecule, that it cannot, in my opinion, cause any nephrocytic effects in the people. But still we need to do animal studies to figure out whether at this concentration of the gadolinium has any effect on that one or not.
Can you comment whether you can use this AS1411 to actually capture circulating tumor cells and exosomes?
People have used it. There are many publications about AS1411 as a molecule to capture the tumor cells from the blood. and these studies have been done, and because of its unique structure forming the core ropelet, that is, I think Dr. Martin Tooley's lab with some other collaborators, they are doing that, and they are making some glass chips to figure out whether this is possible. But there are 2 publications online. They have done that, and it is being used as a self-capturing molecule because it specifically binds to the tumor cells, not to the healthy cells.
And this AS1411 is commercially available? Or-
Yeah, that is commercially available, but we have our lab has a patent on it, so once you can get it from commercial. The sequence is published. You can get it.
Could you tell me how do you fabricate the gold nanoparticle with the IS-4011 and gadolinium together? How do you do that?
I will ask Rajat to answer that question.
You have to spend 2 months in his lab.
So you take a gold nanoparticle, 4 nanometer size, and you add AS1411 to it. So the key part is to heat it to 90 degrees Celsius and with a certain caustic buffer environment and cool it down, dilute it through that AP25 column, and then add it, and then graduate it slowly to 72 hours, every 3 hours, to slowly adding the PBS salt and sonic it at the same time. So for the 72 hours, you are keep doing it. And the reason is to avoid aggregation. Right? The moment it goes to the salt environment of 0.1 molar onwards, it might aggregate. So it's a very sweet spot. And once you're done like for 5-6 months, it's stable.
So after the salt concentration, it will be precipitated?
No. The gold, gadolinium, AS1411 in growth system doesn't precipitate. Well, if we don't put oligonucleotide on it and we just have gadolinium on it, it precipitates out after 1 or 2 weeks. But
And there are like many other protocols available online. People have used different, but this is a little bit more unique to our oligonucleotide and structure, which prevents the aggregation of the gold nanoparticles.
Any other question?
Actually, I want to comment on your questions on the gadolinium. The clinical issue of gadolinium actually is not all gadolinium-based contrast agent, MR contrast agent are the same. Those gadolinium contrast agent that got into trouble is due to the chelator not bound tightly to the gadolinium. So the gadolinium got off from the compound easily. So the toxicity actually comes from the gadolinium itself. So right now what they have done is like multi-hands. They have a special chelator that bound the gadolinium very tightly, so that you stay intact in the molecule as it circulates among the blood. So you do your gold nanoparticle with a tight bonding to the gadolinium. Okay, that is the key. Don't let it get out of the gold nanoparticle. Yeah, the toxicity. Well, then the question is, what is the regulatory aspect of the gold nanoparticles?
It is still,
Yeah, can you comment on that?
it's still in pipeline.
Uh,
Yeah, there is.
The FDA hurdle might be.
But there are like 2 or 3 drugs. They are in the clinical trial approved by FDA.
Those are
with
gold nanoparticle
gold
based.
nanoparticle based.
But do they
Or
have
amine
anything
is one
attached
of
to
them.
it? Do they have anything else attached to it?
One has a PSA molecule on it. Other one, I'm not sure. But there are like few of them are in clinical trial.
Yeah. So I guess in the future we will assess that toxicity question
Yeah.
from the gold nanoparticle.
Yeah. And we got a grant in collaboration with bioengineering and culture translation partnership. and we are doing some of the toxicities and to find out where they ended up. That's a big question. When you inject, where they end up, what's the phase?
In other words, I'm more concerned about the toxicity coming from the gold than from the gadolinium.
But gold is like have shown that it is an inert molecule. It does not have much toxicity, and there are many publications there that they have shown that it's a minimum. But it is controversy, so we are trying that to address that issue in our research.
Of course, I'm one of your collaborators. I want to see it successful.
You are a very important part of our collaboration.
I just want the audience here to understand,
Yeah.
okay, that the problems aren't that
Yeah.
simple.
Yeah.
Yeah.
Any more questions? So at the end, we would like to thank again our speaker. You can now start to quiz, and again, we are so sorry that it's a delay for the
So, you need to go.